Hello everyone, my name is Xiaoyi Sun and I am a postdoc at Beijing Normal University. It is my great pleasure to be here today to share my research. The topic of my presentation is mapping neurophysiological subtypes of major depressive disorder using Connectome-based normative models. As we know, major depressive disorder is one of the most severe psychiatric disorders worldwide, and it is affecting over 300 million people. Major depressive disorder may occur from adolescence to adulthood. One of the most critical features of MDD is the high individual heterogeneity among patients. According to the correct diagnostic criteria DSM-5, patients may show at least 5 of these 9 symptoms. That means patients may have varied emotional cognitive symptoms. This clinical diversity brings up a huge challenge for the understanding of neurobiological mechanisms and the disease diagnosis and treatment. However, the underlying substrates remain largely unclear. These observations have recently led to an increased focus on the investigation of neurophysiological subtypes based on multimodal imaging features. For example, the FA from DTI, the gray matter volume from T1, the functional connectivity from resting state functional MRI. These studies have showed many important findings, such as the subtype differences in functional connectivity of deformal, limbic, and subcortical areas. And they also suggest the potential clinical application of neurophysiological subtypes. However, we know that MDD is a pre-connectomic abnormal disorder. Subtyping based on connectome features will provide very important information. However, the previous connectome-based studies have largely ignored the fact that the pre-connectome can change with age and sex. Moreover, most of them didn't validate the results on multisite. So in this study, our scientific question is whether the connectome-based normative model can identify neurophysiological subtypes, and whether these subtypes show different clinical features. To achieve that, in this study, we collected a multi-site imaging dataset. The subjects are from 9 research centers of China, so they share relatively consistent genetic and cultural backgrounds. We performed strict quality control and standardized proposing on this dataset. Finally, we included more than 1,000 MDD patients and healthy controls. The ages of subjects spanning from 11 to 93. For the data analysis, we first calculated the functional connectivity strengths for each brain region by computing the sum of the functional connectivity between a given region and all the other regions, and the side effects were corrected using combat. Based on the FCS values, we first estimate the typical lifespan trajectories of FCS based on the normative model in healthy controls. Then for each patient, the FCS of each brain region were positioned on the normative model to estimate their individual deviation. Then we mapped the heterogeneous individual deviations among MDD patients. Finally, based on patients' individual deviation patterns, we identify the MDD subtypes and validate the subtype differences in baseline clinical features and longitudinal treatment prediction. Here are our results. As I mentioned before, we estimate the normative models for each brain region. We found some regions showed increased age-related FCS change. For example, this is the normative model of post-central gyrus, and some regions show decreased FCS change with age. For example, this is the normative model of posterior cingulate cortex. For the individual deviations among MDD patients, we calculated the number of extremely deviated regions for each patient. We found more than 17% of patients showed extreme deviations in at least one brain region. We also calculated the percentage of patients who had an extreme deviations in each brain region. We found for any single brain region, no more than 4% of patients extremely deviated from the normative range. These findings suggest that while alterations in FCS exist in most of MDD patients, the specific brain regions affected by these alterations vary markedly across patients. Based on patients' individual deviation patterns, we clustered patients into two subtypes. And we can see that there is no significant site differences in the number of patients within each subtype, and the overlap rates between leave one set out validation and May results were all higher than 92%. That means the subtyping results showed a high intersite reproducibility. We found subtype 1 showed positive deviations in the deformed limbic and subcortical areas, and negative deviations in the sensomotor and attention areas, while subtype 2 showed a reversed deviation pattern. The deviations of subtype 1 were more severe than subtype 2. We validated subtype differences in demographic and clinical features. The severe deviation subtype, which showed positive deviations in default mode, was significantly older, had a higher medicated proportion, had more severe symptoms in the suicide item, while the moderate deviation subtype showed more severe symptoms in the work and activities and depressed mood items. Importantly, we also included a subsample of patients who received 6 months treatment. We used the baseline deviations to predict the treatment outcome at each subtype by SVR method. We found the baseline deviation patterns can predict treatment outcomes for the severe deviation subtype, but not the moderate deviation subtype. Finally, we also validate our findings under different analytical choices, and we found our results are very stable. Okay, that's all my results. In conclusion, we observed great heterogeneity in functional connectome deviations in MDD patients, inspiring the identification of two reproducible neurobiological subtypes, which differed in imaging and clinical features and treatment outcome prediction. These findings shed light on our understanding of different neurobiological mechanisms underlying the clinical heterogeneity of MDD, and essential for developing personalized treatments for this disorder. By cooperating with clinical doctors, we are going to investigate the potential clinical application of neurophysiological subtypes on disease treatment, such as using different parameters and strategies for different subtypes in TMS treatment. In the end, I would like to thank my supervisors Mingwei Xia and Yonghe, and all the DIDA MDD group members for their contributions of data collection and suggestions. That's all for my presentation. Thanks for your attention.